All right. So as I was saying at the start of class, Ms. Quigley and I have decided to put a. Excuse me. Let me deal with that. All right. Sorry. So as I was saying, Ms. Quigley and I decided to switch gears, and we are going to be switching from the genetic variation topic into the biological investigations. Our thought process with that was that you guys are all over the show with how successful you've been with remote learning. And since you guys are all over the show with your remote learning, it's really hard to catch everyone else up. I'll wait till you guys are done talking. So, where's my train of thought? We decided to put a pause on the genetic variation. So with that genetic variation, we're mindful everyone's in a different stage in their learning. And we want to give you guys, one, time to play catch up. So by stopping this topic, it means that people have a chance to play catch up. And two, it also gives you guys an opportunity to start fresh. And so that way everyone's on the same playing field. Because it's really hard for us as teachers to teach you guys when you're in such different places. So that's why we want to start fresh, so that way everyone's hopefully on the same page. Does that sound like a good plan? Yeah. All right, so today what we're going to be doing then is basically a lot of introduction with that genetic variation talk, or sorry, not genetic variation, the biological investigations. We're going to be revising the scientific method because this whole topic is all about looking at investigations. So that's what we're thinking about for today's lesson. Cool. I won't answer the do now because we will go through that stuff when we're doing the notes. Does that sound like a good plan? All right. Cool. So, lesson plan for today. The first thing we're going to do is some information on the standards so you guys know what to expect, and then I'll do some notes on the scientific method to review, and then you guys will have a chance to practice that as well. Cool? All right. I don't have the checklist created yet. And that's because we have decided last minute to change. And so next lesson, I'll hopefully have some more resources for you guys. But for today's lesson, I think we'll be okay without all that sort of stuff. Cool? All right, let's talk about what this standard is about. So first off, this is Bio 2.1. And it is an experiment-based assessment. So what you guys will be doing is you are going to be conducting an experiment that's biology related to answer a biology question. Yes, very exciting. Do you guys remember the physics experiment you guys did last year when you rolled the ball? I think you're rolling loo roll or something like that because you guys did it in lockdown. It's very similar to that assessment, if that helps. So with that assessment, it is in two parts. The first part is a practical component where you guys will be working in pairs to collect data. And that's really nice, and it helps ease off the pressure from the assessment because you can work with somebody else and plan. The second part of the assessment then is the write-up of the practical report. So that's explaining the results, doing a graph, that sort of thing. The second part needs to be done individually. So just like when you guys did the roll the ball or roll the loo roll experiment, it's very similar to that. Are we good so far? Does that hopefully ring a bell from last year? So it's similar to that, which is the good news. Like I said, it's an internal. It's going to be worth 4 credits. There's also a numeracy component, so some of you guys may not have gotten your numeracy just yet. I think most all of you guys have. But this gives you a chance to get some numeracy evidence as well. Are we good so far? All right. Cool. Let's talk about the scientific method and the standard so you guys know the context of this kind of work. Because one, I haven't had a chance to make the booklet, but two, I think it'd just be good for us to try to see how you guys like it as a note forming or way for me to give you guys notes. You might prefer this to what I've been doing previously. Yeah, so you should be writing this down. I'm happy if you want to type it. I'm happy if you want to handwrite it. I'm not really fussed. I handwrite it because it slows me down. Because I'm a very fast typer. So this helps me kind of make sure I don't go too fast. Are we good so far? All right. So let's talk about what investigation scientific method is all about. The first thing we need to think about when we're doing investigation is thinking about the purpose. So the purpose is basically our reason. Don't worry, I will scan all these notes and I will put them online if you guys need them. So for this, we need two separate things to think about in the purpose. Basically, the first thing you need to make sure that we have in our report is our aim. So that's explaining what we're doing and why we're doing it. The second thing you're going to need to have is your hypothesis, which is our prediction. Are we good so far with that? So that's the first thing we're going to be thinking of, is the purpose of that experiment. Now, the nice thing is we will give you guys the purpose in a sense of we'll tell you what experiment we're doing, and you guys will write the aim and the hypothesis for it. Once we do that, we need to think about design. So that's the next step. Now, with that design, there's several things that we need to think about. The first thing we need to think about when we're thinking about design is how do we create a fair test? So we need to make sure whatever experiment that we carry out is, something that's valid and something that I know I can trust the results. So for me to be able to trust the results, I need to make sure that I've done things to make sure I have a good experiment. So some things we need to think about when we're doing that is the independent variable. I often will abbreviate independent variable as IV. Now the independent variable, can we put away the phone? Thanks. I don't know why we think just because it's the third session we're not doing anything. Come on, get your notes. Put away the phone, take out the headphones. I can wait. Headphones. Put it away. I don't want to see the phone on the desk. And I want you to have your notes out. Where's your notes? You're waiting for what? There we go. All right, so the independent variable, that is the one that we are changing. It's basically what that experiment is about. The way I like to remember it is that it is independent. We can change it. Independent means free. What else did I want to add about that? How many independent variables are we allowed to have? A lot? We're only allowed one. So what we change, you can only have one. The reason why we can only have one is because if I change too many things at once, I then don't know which one had the effect. So let's say I was trying to bake a cake and I want to make the cake really, really tasty. If I change the butter content and I change the sugar content at the same time and the cake is really mucked up and doesn't taste good, I don't know if it's because I've added too much butter or if I added too much sugar. So that's why we change one thing at a time, so that way I know whatever the result is, I know what was the effect of that. Cool? So the first thing we think about is that independent variable. The second thing we need to think about is our dependent variable. Dependent. I often abbreviate that as DV. So that is what we are measuring. What we measure in that experiment. Can I have more than one more than one dependent variable? Yes, I can. So this one we are allowed to have more, and it's often that you will have more than one because you want to measure multiple things. In the context of this experiment, we will only measure one thing. But in general, we can have more. Can have multiple, but in this case, we will only have one. So just so you guys know as context. All right, cool. So what else did I want to add in there? The last thing we have to think about then are control variables. All right, so control variables are things that I need to keep the same. Put sample, sorry, same. So I need to make sure those things stay the same so that way my experiment is a fair test. So like, for example, when I was talking about that cake, I can only change one thing at a time. So let's say I change the amount of butter I use for that cake. Everything else between my different cake experiments, the sugar, the flour, the baking soda, all of that, the baking time, the temperature, those all need to stay the same because those are my control variables. Are we okay with that so far? All right. So with all that information, when I think about the independent variable, the dependent variable, the control variables, what makes things a fair test, we take all that information and what we design from there is my method. And actually, I might change colors because that is kind of a new idea. So we are thinking about our method. So the method is something else that you will also need to be writing, and that is your set of instructions. Now with that set of instructions, I want you to write them as, step number 1, blah, 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 step number 2, blah, 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 step number 3, blah, 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 etc., etc. Are we okay with that so far? Cool? All right, I'm gonna move this. You okay? All right. In a good method, we should first think about the range. For the independent variable. So when I say a good range, I mean I need to have numbers that would give me good data. I don't want really small numbers that are close together because it may not give me the full story. So we want a good spread. But at the same time, we don't want a huge, huge spread that I'm going to get problems. So when we think about range, we think about basically an upper and lower limit. So what would be a good idea for that experiment as like a maximum value to test that isn't too expensive? And then what is a lower limit that we can test that isn't going to make my experiment run super, super slowly or not show me any results? So that's going to be dependent on the context of what we are doing. The other thing we need to think about is sampling bias. So basically, when we are doing some experiments, could there be something with my measurements that could affect and influence my results? The other thing to think about is errors. So when you guys carry out your experiment, you might make mistakes, and it's important to note what those mistakes are. I should say checking for errors. Are we good so far with that? Does that all kind of ring a bell? You guys should have been writing a method last time. So that method is something that, again, you guys are going to need to be writing. We're not going to give it to you, but we will show you an example. So we will explain what the experiment is, but you guys then have that freedom to think about, okay, with my experiment, what do I want to do? Okay, so we give you the guidelines, and then just like you did last year, you decided what you wanted to have as the height of the toilet roll rolling down. All right, cool. After we have that data, the next thing that we're going to do is record and process it. So that's our next section, is record and process. So the first thing we have to really kind of think about with that record and process is we have raw data. So you do that experiment and you collect it. In particular, what you guys will need to do is make a table from that. So make a table. When we think about that raw data, you should have 3 trials that you then average. The other thing you should then do after you have those 3 trials and then average it is make a graph. You guys should have done that hopefully last year with that the roll the ball experiment. Now, when you make a graph, you also have to be mindful about which graph you use because there's 2 types. There's the bar graph and there's the line graph. Does this hopefully ring a bell? Bar graph kind of looked like that with the little bars. Yeah? And then line graph was the one with the little dots that you maybe did a line of best fit. Yeah? With the bar graph, you use the bar graph when you have categories of data. Categories of data. So for example, if I had like favorite animals, and then I had a poll to see which one was the favorite animal. With the line graph, you have 2 sets of number data. So say, for example, the height of the ramp versus how far the ball rolled. Does that ring a bell? Both of those are measurable, whereas one is category and measurable. Let me know if you need me to slow down or clarify anything. We good so far? All right. Cool. So that's some recording and then processing. The second part then of your processing is doing things like writing a conclusion, writing your discussion, writing your evaluation. So first thing when we're doing that processing is a conclusion. So that's basically like your little summary of what happened. So when you're writing your summary of your results, what you're thinking about is, first off, you're just making it's basically like 2 main chunks when it comes to this year. The first thing is just your key sentence, which is as, and then you state your independent variable, and then you state what you did. So maybe you increased it, maybe you decreased it. So as I increased the height, then you go, then the, and then you state what the dependent variable is and what happened. What happened to it? So as, say, for example, I increased the height of the ramp, then the dependent variable, so in that case, the distance the ball rolled increased. What you then think about, and that's kind of what's going to be a little bit of an extension for this year, is that you need to link it back to the hypothesis. So was your hypothesis correct? And then after you state whether or not that hypothesis was correct, you want to compare it to other data. as your further evidence. So when we say other data, you might be comparing it to what your classmates have got. You might also be comparing against data that I've given you. So I will depending on the assessment and depending on if we're doing some practice learning in class or you're doing the actual assessment, there will be data that's given to you that will be similar but not identical to what you guys did. And so what you're going to be able to do is compare it and say, you know, I can't trust my results because when I look at this data, it has the same sort of pattern, sort of thing. If your results are different, then you can be like, well, there must be something wrong with my experiment because it's not matching other data that I see that's available. Does that make sense so far? It's like real general kind of what we expect from an experiment. All right. Huh?
This reminds me of the science fair.
The science fair. It basically is, because that's what you guys are doing as an experiment. Cool. Can I move it? No? All right. But that's basically what this is. It's like an experiment for the science fair. Except you guys don't have that creativity of choosing your experiment. I'm going to tell you what your experiment is. All right, move it. All right, so the other things that we're going to be thinking about is once I have those results, I need to explain why I got those results. So we're going to be thinking about two things. The first thing is going to be your discussion. So that is your explanation. So explaining the results with science ideas. Or concepts, I should say. Concepts, content, that sort of stuff. So like, again, I use that roll the ball experiment a lot because you guys have done that one and you hopefully have that prior knowledge. When you guys were explaining why that paper towel, the toilet paper rolled further, you were linking back into gravitational potential energy and how the higher the ramp, the more energy it had to then go further. In this context, there's lots of different experiments we can do because this is a biological idea. We have chosen as our biological idea for us in this topic, we are going to do osmosis. So osmosis is going to be the scientific theory that we look at in the context for this standard. Now, when you guys read up on the standard, there's going to be lots of different examples. Some use photosynthesis as their science ideas. Some use, can't remember, I don't think it was snails, it was something else. But that's what we're thinking about in this case. The nice thing about pausing the genetic variation where we have paused it is that we have talked about cells because we were talking about mitosis and meiosis. So with that meiosis and the cells, it ties well into osmosis because with osmosis, we're also going to be talking about cells. And we're going to talk about cell membrane. So it's a really good kind of link, and that's what Ms. Quigley and I decided right now would be a good time to kind of pause genetic variation because you guys have just wrapped up on cells, and this topic looks at cells as well. Cool? So if you guys want, you can start reading up on osmosis. You can start learning about diffusion and the cell membrane because that all ties into the science content for this topic. Are we good so far? And then the last thing we do is our evaluation. So evaluation is just thinking about, did we have a fair test? Evaluation of your experiment. So with that evaluation, we're going back to thinking about, where is it? The method and checking for errors, thinking about our range. Was our range actually good enough for our upper and lower limits? Thinking about back to our variables. Was my control variables all accounted for? And that's, again, specific to what you guys did. So we will give you the scaffolding of the experiment and what to do. You guys then take it. You will write your own method, carry out your own experiment, and you can reflect on how good or bad you've done it. So, the important thing to remember is you can have a rubbish experiment, but that will not prevent you from getting a merit or excellence. You can fix the rubbish experiment by having a good evaluation. So if you're able to tell me what was wrong with that experiment, even though you have bad data, I can still give you higher marks. Does that make sense? All right? So if you can explain to me why your experiment was bad, it can save your assessment grade. Are we good? Can I move on? Can I move this? All right. So that's basically the notes from me, and then I want you guys to have a chance just to practice the scientific method with some example data. What do you guys think about me doing the notes like this on paper instead of just doing a PowerPoint? Do you prefer this? Okay, cool. That's good to know. So I'm happy to give our notes like this because I know it slows me down. All right. So, by looking at the scientific method. So, what I've done is I've provided some Google Slides and some Google Sheet documents, which have some data. And from that, you guys will be able to practice that scientific method. So let me show you where it is. Cool. So level 2 biology, lesson number 1 in the scientific method. I have created some Google Slides and a Google Sheet. So let's first look at those Google Slides. Cool. So, I'm happy if you guys want to work in groups, or you can work individually or in pairs to get this done. First off, I've explained some context because you're going to need that in order to understand the experiment that they've carried out. So this example was an experiment based off of photosynthesis. So photosynthesis is that process of taking light energy and converting it into chemical energy, sugar. So we have carbon dioxide, we have water, and with that light energy, we're able to make sugar and oxygen. Yeah, sugar. Yes, it is on Google Classroom. All right, so that is the general kind of definition for photosynthesis. What I need you guys to do is use this data table here and think about what is the independent variable, what is the dependent variable. So when you look at this, what do you think, which one's which? Write it in. Then I have a little experimental setup. So with this experiment, they had a light source and they were heating up the water, and they have all the different temperatures, and they were measuring the bubbles that were coming off. So you were looking and measuring it. This is one example of what we could do as a biological investigation. We're not doing this one, so that's why I'm using it. I want you to write an aim. I want you to write a hypothesis and what you think would happen. And I want you guys to write control variables. So that's the first half of what I want you guys to do. When you have that done, call me over. I'll check it, and if I'll give you a sticker for it. The second half is processing data. So, gentlemen, with that processing data, I've made a Google Sheet. I want you guys to see what you guys can figure out. I want you to average, and I want you to create a graph. So average, create a graph, and insert the graph here. And then the last step is your conclusion. So 2 stickers for getting this work done, 1 for getting up to this point, and then the second one is the graph and the conclusion. Does that make sense? You guys have the rest of the period to get this done. I will walk around and help. And like I said, you have data to help you kind of figure out which one's which. But I just want to see what you guys can kind of pull from that table when it comes to the scientific method. All right, I'll leave that up for you guys. Phones if you guys need to borrow any. Cool. Okay, I just want to wrap up today's lesson. So with the Google Doc, if you haven't finished it, then that's basically going to be your homework to finish the Google Slides and the Google Sheets. In our next lesson, I'll go over Some of you guys have already seen some of the little tricks with how to average it very quickly. I'll also go over how to kind of do some of that graphing stuff so you guys know what to do. So to summarize what we worked on today, we were going over the scientific method and we were relating it into specifically what you're going to be doing this year for the standard. By the end of today, you guys should be able to carry out the steps of the scientific method. We will practice this obviously multiple times. And you guys should hopefully be able to identify that independent, dependent, and control variables. Cool. So you guys are the last period of the day, so 3 things need to happen. First thing is I need all the tables to be wiped down. So on the bench next to the door, there's spray and there's wipes. Second thing that needs to be done is the chairs need to be put up. The chairs can only be put up once the tables are wiped. And the third thing is I need all of my windows closed. So we're not leaving till those 3 things are done.